What is a cell? Part 1. What does it mean if we call a cell or describe a cell as being prokaryotic versus eukaryotic? Prokaryote means before the kernel. The kernel or seed is referring to a structure that we see in other types of cells we call eukaryotic, called a nucleus. Well, prokaryotic cells have no nuclei. And this is the fundamental, huge difference between a prokaryote and a eukaryote. The absence or the presence of a nucleus. An important, unique component that's found in prokaryotes is a structure called a cell wall. The cell wall of bacteria is composed of peptidoglycan. Archaea have cell walls made of pseudopeptidoglycan. Now, the peptidoglycan thickness or thinness will tell us what kind of staining quality we will see when we take these microorganisms into the laboratory and stain them. If we say a prokaryotic bacteria is gram-positive, what does that mean? Well, in truth, it tells us a good deal about the cell wall composition and some of the other unique characteristics of that kind of prokaryotic bacteria. Organisms that have very thick peptidoglycan layers composing or making up their cell walls are often going to retain the purple color that we use when we perform a gram stain technique. That very thick peptidoglycan layer entraps most of the purple color, or crystal violet. The mordant that we will use in the second step of the gram stain procedure will stabilize that purple color so that when we go through the third step, which is decolorization, most of the purple color is retained in that very thick peptidoglycan layer. Now, it doesn't mean that it's permanently etched or permanently embedded in that thick peptidoglycan layer. It simply means it's more difficult to decolorize or remove that very saturated purple from that very thick peptidoglycan layer. And of course, that thick peptidoglycan layer gives that type of bacteria a nice, sturdy covering, so it is well protected from the outside environment. If instead of a gram-positive organism, we run through our gram stain procedure and let's say all of the purple color comes out, there's a good chance you have what's called a gram-negative bacterium or prokaryote. This simply means that with a gram-negative cell wall, you find a very, very thin layer of peptidoglycan sandwiched between two membrane structures. Two biological membranes. One's called the cell membrane, which is the innermost membrane, and one called the outer membrane on the outside surface of the peptidoglycan layer. The outer membrane is a rich source of a chemical called lipopolysaccharide. Lipopolysaccharide, also abbreviated LPS, is, as the name implies, a composite of two biochemicals that we call lipids and polysaccharides. The lipids would mean essentially a chemical that's very much like an oil or a wax, something slippery of that nature. The peptidoglycan is right underneath that LPS layer, well protected by the outer membrane and the lipopolysaccharide. Lipopolysaccharide, or LPS, is a unique component of gram-negative bacteria, and in fact is the cause of the symptoms that we see in septic shock. LPS has the lipid component called lipid A, remember that slippery or sort of waxy, oily material that we find in LPS, or lipopolysaccharide, and it's the lipid A of LPS that triggers the symptoms of septic shock. It's really the toxic or nasty portion. So it's simply part of the structure of the cell that triggers some of the symptoms that we see when someone has a very serious infection. Now, the fact that it has a very thin peptidoglycan layer, even though you can see that the membrane looks a lot thicker, The membrane of that thin peptidoglycan layer is not sufficient to trap and retain a lot of the purple color. So when we go through the decolorization step of the gram stain, virtually all of the purple crystal violet is decolorized or removed. And then when we add our counter stain or the secondary stain that we call safranin, the color of the safranin is retained in that very thin peptidoglycan layer of the gram-negative bacteria. Here's a close-up view of LPS. LPS, located in the outer membrane of gram-negative bacteria, is in fact the source of lipid A, which is the toxic bit that triggers symptoms that we associate with septic shock. These are often sources of exam questions. It's important, especially for us as healthcare consumers, to understand that if we have an infection with a gram-negative bacterium, that sometimes we can have very serious outcome or serious problems if that infection isn't contained or isn't controlled, and we develop symptoms and signs of shock. It's this chemical from the bacteria that's triggering that drop of blood pressure, which is what we associate with shock or hypotension. So please make sure you understand what the abbreviation LPS means, that's the abbreviation for lipopolysaccharide. Where is it located? In the outer membrane of gram-negative bacteria. And what part is toxic? The lipid A, which often triggers the symptoms we associate with septic shock. Eukaryotic cells have true nuclei, and that's what the U means, true, karyo, seed or kernel. So, eukaryotic cells, the biggest difference between pro and eukaryotic cells is the presence of a nucleus in a eukaryotic cell. Plant, animal, fungi, protist, algae, all have eukaryotic cell structure. If you take a look at this generalized eukaryotic cell, you can also see inside of the cell, there's lots of other things going on. All kinds of other bits and pieces that we associate with eukaryotic cells, in addition to the primary structure that's really noticeable, and that's the nucleus. Eukaryotic cells that have cell walls, not all of them do, but if we do have a eukaryotic cell from an organism that has cell walls, if it's a plant-derived kind of organism, we'd expect to find cellulose. If it's another type of organism like a fungi, we might expect to find chitin. Both of these materials, cellulose and chitin, are very sturdy structures and biochemicals to be used in eukaryotic cells. When we look in a prokaryotic cell, the genetic material or the hereditary blueprint is found in the form of a chromosome that's a single circular chromosome. So a little ring of DNA that's called the nucleoid. Now if we look inside of a eukaryotic cell, we do find a true nucleus, and inside of that eukaryotic nucleus will be chromosomes, multiple chromosomes, more than one, and they tend to be linear or like little sausages. Both pro and eukaryotic cells may have spores. The fundamental difference would be what kind of spores. In prokaryotes, particularly bacteria, we find structures that we call endospores. And I do make a distinction between bacterial endospores that we can find in prokaryotic bacteria versus vegetative spores that we find produced by certain types of fungi. Bacterial endospores are very sturdy structures. They actually can resist heat, desiccation or drying, extreme temperature changes, freezing, acids, alkalis. They're just very tough. little structures. The endospores from bacteria contain a complete copy of all the genetic material. But think of it as a little life capsule or a little time capsule. Everything's in that little spore, and when the conditions are just right, that spore will land on a nice new place to grow. And the word that we use when the endospore essentially starts to grow is germinate. And actually, the word, like we talk about seeds, even though spores are not seeds, we say that the spores germinate and eventually start to produce fresh, new bacterial cells. If we look at a prokaryote, we also find very few membrane-bound structures that we call organelles. So typically, prokaryotic cells don't have organelles, but if we look inside of a typical plant or animal cell or other kind of eukaryotic cells do have internal organelles, such as mitochondria, chloroplasts, lysosomes, peroxisomes, Golgi apparatus. Lots of structures and things that we see in a eukaryotic cell, all kinds of busy little things that are lacking or do not appear in a prokaryotic cell. Now, the vegetative fungal spores that we might see produced by certain types of fungi are very sensitive to desiccation, acids, alkalis, heat, and cold. Those are things that will actually destroy the vegetative fungal spores.